Welcome to Active Motif's webinar on spike-in normalization for NGS-based assays entitled Spike-in Methods for ChIP-Seq, ATAC-Seq, CUT&RUN, and CUT&Tag it. My name's Brian Egan, and I lead the R&D team here at Active Motif. In today's webinar, we're going to review our old spike-in normalization method for ChIP-Seq, and then spend some time on spike-in methods for 4 more recent assays. Those are CUT&Tag, ATAC-Seq, CUT&RUN, and a CUT&Tag based method for R-loop detection. We'll then talk about the need for spike-in when using negative controls and other experimental settings where spike-in is needed. I'm going to start by revisiting the ChIP-Seq spike-in method that we published a few years ago. At the time, we were doing ChIP-Seq services for a number of pharmaceutical companies. All of them were doing work with developing EZH2 inhibitors. So all of them were seeing global reductions in K27 trimethylation, but when we did the ChIP-Seq experiments, we didn't see that expected result. So this is an excerpt from the paper we published in 2016. In this case, we were using CPI 360, which was an EZH2 inhibitor from Constellation Pharmaceuticals. And you can see by Western blot that after 4 days of treatment, you got a global reduction in K27 trimethyl, and even a further reduction at 8 days treatment. The control or loading control was a antibody for histone H3, so this is total histone, so it's not affecting total histone level and specific to this particular modification. But the problem is when we did the ChIP-Seq analysis using an antibody against H3 K27 trimethylation, we didn't see the expected reduction in K27 trimethyl ChIP-Seq signal. So based on these unexpected results, we started working on a spike-in method as a possible solution. And the method we came up with is in a schematic here. You use your test chromatin, in this case human chromatin, along with an antibody against your target of interest, in this case H3 K27 trimethylation. And in the same reaction, we add Drosophila chromatin and a Drosophila specific antibody. And in this case, H2AV, so this is a Drosophila specific variant. Now, in theory, we could have left this antibody out and relied on cross-reactivity of the H3 K27 trimethyl antibody to the Drosophila modifications. But we wanted to make this approach universal, so we chose to add a second antibody. So, if we were to do these types of experiments looking at transcription factors, where those transcription factors aren't well conserved across species, this antibody gives us the flexibility to be able to pull out Drosophila chromatin in a very specific way. So in this method, we add all these into a single tube. We perform ChIP as we would normally, make the library sequence, and then map those sequencing reads to the test genome or to the Drosophila genome. And then we count those Drosophila reads and make correction ratios to normalize the human data. So again, from the paper, if we look at Drosophila counts in untreated samples, we get a number of counts. If we look at counts from the treated samples, we actually get more Drosophila counts and even more at the 8-day treatment. So these differences then are used to make a normalization ratio. And then that normalization ratio is applied to the ChIP-Seq data, and we essentially can use those ratios to downsample the human tag counts. And when we do that, we now see this expected global reduction in K27 trimethylation. One other note here is we typically strive from 2 to 10% Drosophila reads. We want enough reads to be statistically meaningful, but we don't want to have so many reads that we overwhelm the system and waste a lot of sequencing reads on the Drosophila portion of the experiment. So again, this was published. I won't really spend much more time on this, but it was published, and you can go into that publication, look at more detail if you're interested. So now that there's other methods of epigenetic methods to look at epigenetic effects genome-wide, we were curious to know whether those methods also require a spike in normalization. And so we started with cut and tag, and in this example, rather than using a treatment that changes a histone modification globally, we used this an experimental model where we're using cell titration. So essentially, we were mimicking more H3K27 trimethylation by just adding more cells. And so we did the experiment, and as we saw for ChIP-Seq, we actually don't see any major differences in peak intensity when we use 200,000 cells all the way down to 50,000 cells. So we suspect that a normalization is needed to able to see these differences between different cell numbers. And so we did design a new spike-in, and our new spike-in is to use Drosophila nuclei. So when we were doing ChIP-Seq, we used Drosophila chromatin because that's the input material for ChIP-Seq. And CUT&Tag cells or nuclei are the input material. So we chose to use a Drosophila nuclei. And so this is a design of our, or a schematic of our cell titration model. This was actually taken from a 2017 paper that used Drosophila nuclei to normalize RNA-seq data. So the idea is that you start with different numbers of cells and a constant amount of Drosophila nuclei. And that's represented in this graph where you have increased numbers of cells in white and the constant amount of Drosophila represented in purple. And in this case, we used 10,000 Drosophila nuclei. So after you sequence, all your samples get the same amount of sequencing reads. So again, that's represented in white, and your Drosophila reads are represented in purple. So you can see in the first sample, you have proportionally more Drosophila reads because proportionally that's what you added. These ratios then are used to normalize the data. So when you normalize that purple bar, you then normalize the human reads, and that becomes representative of your starting material. So it's similar to the inhibitor treatment, but again, using cell numbers instead of drug-induced global changes. So with that system in place, the cut and tag experiment works pretty much like it would normally. You start with cells, you mix your Drosophila nuclei into the same exact sample. You add your antibody of interest. You also add the H2AV antibody at the same time. Then you use add the secondary antibody, then the protein A TN5. You get tagmentation around the target antibody, and then you can amplify that sample and sequence it. So this is the model that we used. Again, I just showed this data without spike-in normalization. If we do H3K27 trimethyl cut and tag, we don't see differences across the different cell numbers from 50,000 to 200,000. Once we apply the spike-in normalization, we now see a stepwise decrease in signal intensity as we lower the cells. So this shows us that the spike-in normalization actually does work for cut and tag. We then went back to our EZH2 inhibitor model just to show that our cell titration model was relevant. Again, here we used the GSK125, which is a different EZH2 inhibitor. We did H3K27 trimethyl cut and tag, and without normalization, we don't see the expected decrease in EZH2, or I'm sorry, in H3K27 trimethylation. With the spike-in, we see this dramatic global reduction in H3K27 trimethylation. Once we were able to show that spike-in method was relevant and worked properly with cut and tag, we tested similar methods with 3 other mapping technologies. And so there's with cut and run, our cut and tag based R-loop method, and ATAC-Seq. So for all of these new methods, we also use Drosophila nuclei as a spike-in. For cut and run and R-loop, we also use the H2AV antibody, just like cut and tag. But for ATAC-Seq, since this is an antibody-independent method, we don't have to use the H2AV antibody. And so I have the spike-in results for each of those methods in this slide and the following slides. So for cut and run, we used an EZH2 inhibitor. Instead of using antibodies to H3K27 trimethyl, we used an antibody to EED. So EED is a protein that's in the polycomb complex. So EED directs binding of the polycomb to H3K27 trimethyl. So in the presence of an EZH2 inhibitor, we expect a decrease in EED binding. So again, without the spike in, we don't see that expected decrease. And with the spike in, we see a reduction in EED binding. On the right, we are looking at R loops. So R loops are genomic structures where you have 3 nucleic acids. First of all, you have an RNA-DNA hybrid, and that's in conjunction with a single-stranded DNA piece. And so these structures in the genome regulate gene expression. Just so happens that there's an antibody available against DNA-RNA hybrids. It's called the S9.6 antibody. So we can use this antibody to direct cut and tag to R loops and get an R loop profile. It's essentially the same as a regular cut and tag, except you're swapping out the antibody. So no one's really ever looked at R loops in the context of an EZH2 inhibitor, so it's really not known what would happen. We theorized that with EZH2 inhibitor treatment, you get less heterochromatin, which means you should get more open chromatin and more transcription. So we might expect that you would get an increase in R loop signal. So when we apply our spike in, or when we do this experiment with an EZH2 inhibitor, we don't see any real changes in the intensity of the peaks for R loops. Once we do the normalization, we actually do see an increase in R loop signal, which is what we hypothesized. So, so I did want to note that we only did this experiment one time, so I don't want to comment too much on the biology, but in this one experiment, it performed as we had hoped. And then lastly, we also did a spike in with ATAC-Seq. So again, we just set up our normal ATAC-Seq reaction. We add Drosophila nuclei, and the TN5 integrates into the test sample and into the Drosophila genome. And we make normalization ratios based on Drosophila tag counts and to normalize our test, our sample. So in this experiment, we went from 10,000 cells up to 200,000 cells. Without spike in, you don't see any major differences in signal intensity across those cell numbers. And after spike in, you see a stepwise decrease in signal intensity, demonstrating that we can also use Drosophila nuclei spike in method in the ATAC-Seq assay. In the next section, I want to show a couple surprising examples where spike in applies to negative controls. And in the first example I'll show, we're going to look at IgG as a negative control in cut and tag. And in the second example, I'm going to show an RNase treatment as a negative control for our cut and tag based R loop system. So this is an experiment where we looked at H3K4ME3 with cut and tag and our negative control antibodies IgG. Once we amplified the libraries, we ran them on a tape station, and we see exactly what you would expect to see is that you have dramatically more library from your H3K4ME3 test sample than your negative control IgG sample. And in this case, it's about 30-fold difference, 34-fold more library from H3K4ME3. Once we sequence that data, here's an IgV screenshot, we see that we're actually getting peaks in our IgG control. So this was puzzling, and we thought about it a while and realized that, when you're sequencing next-gen libraries, it's standard practice to put exactly the same amount of library from each sample into your sequencing run. And so when you do this, you actually artificially add more of this library than this library. So we essentially add 30 times more of this library than this library to the sequencing run, which artificially boost the signal. And so we thought that a spike in normalization may rectify that problem. And so we did do this with normalization, and after normalization, you see that we get signal from H3K4ME3, and now we don't really get peaks in our negative control IgG. Similarly, with the R-loop assay, a good negative control for the R-loop assay is to treat the cells with RNase H before doing the CUT&Tag experiment. So the idea is that you have this DNA-RNA hybrid within the genome, and RNase will cut that hybrid and essentially destroy the R-loop. So you shouldn't really be getting a targeted tegmentation to those locations anymore. And so again, when we do the experiment and run the samples on a tape station, we get what we would expect. So dramatically less yield from our RNase treated sample compared to the normal sample. However, when we do the sequencing, map it, and create these IGV files, we actually see that we're still getting peaks in the RNase sample. When we add the spike in normalization, we now selectively decrease peak intensity from the RNase. So essentially, there's no peaks left. So again, these are the IGV screenshots, but you can get a feel for the entire data set with the meta-analysis, where you can't see clear separations of the the RNase and the 9.6 samples here. And after normalization, you get a clear separation of the negative control RNase treated samples compared to the normal samples. And so, I think the other thing to point out here is you ask the question, why are your peaks located in the same regions as H3K4 ME3 here and in R loops here? And the reason for that is that in a CUT&Tag assay, you always get spurious tegmentation, and that spurious tegmentation occurs in open chromatin regions. And so the amount of tegmentation is minor, but it still occurs in those regions. And then once you amplify up the material, you see peaks in those regions. Well, those open chromatin regions would overlap with ATAC-Seq data. Of course, ATAC-Seq data overlaps with the promoters of H3K4ME3 marked genes. And then R loops also control gene expression are basically in the promoters of most transcribed genes. And so that's why we think we're seeing peaks in the same locations. So with that, I just wanted to share when we think it's appropriate to use spike-in controls. The first one's obvious, and that's when you do a drug treatment with a epigenetic inhibitor. So that probably applies to any epigenetic inhibitor, but we've shown specifically over and over again that it's absolutely necessary when you do H3K27 trimethylation in the presence of an EZH2 inhibitor. And so, another example might be when you're doing different cell types and you want to compare different cell types. There are circumstances where you have different levels of different histone PTMs in different cell types. And so without the spike-in normalization, you wouldn't really be able to see those global differences in histone PTMs across different cell types. There may be instances where you have different cell numbers at the beginning of your experiment. So maybe when you counted the cells, there were errors in counting. Maybe you have treatments that are toxic and kill the cells. Maybe you're doing an experiment where you're using primary cells and you're just not able to get enough of some of the samples. I think in all those cases, a normalization would be able to correct for any of those differences in starting cell numbers. And then the last one is from the data I just showed. And in this case, when you're using negative controls, it seems necessary in a CUT&Tag based assay, at least, to use a spike-in normalization. And before I close, I just also wanted to comment on other available normalization methods. There are other groups that have done normalization in CUT&Tag and CUT&RUN. In particular, for CUT&RUN, the original publication showed that there is contaminating DNA in the TN5 MNase preparations, and that contaminating DNA is carried through the assay, and it can be used as a normalization. And I would just want to caution against doing that. First, it can be problematic because each preparation of PAG MNase is going to contain different amounts of contaminating E. coli sequence. And so it's probably not going to allow you to have consistency across experiments that occur over large periods of time. And so the other thing is that, you know, especially if you're using commercial PAG TN5, that you may not be getting the E. coli contamination in those preps. So you may be relying on that for normalization. It's simply not present. And then lastly, I think for E. coli DNA, it's really not going to work for cut and tag because it essentially will get washed away during the washes for cut and tag. So I just wanted to point out that, you know, although there are other spike-in methods available, there's some caveats that go along with using them. And then in summary, you know, I did share our spike-in method for ChIP-Seq. That's been around for a number of years. We use it in our services business. We also sell a product for ChIP-Seq spike-in. The new data I shared today allows spike-in normalization for ATAC-Seq, CUT&Tag, and CUT&RUN, and all those new methods are available at Active Motif along with the corresponding Drosophila nuclei and antibodies for spike-in normalization.